All right, to finish up, we're going to talk about what happens when there's mutations. We're also going to talk about DNA repair because luckily there are ways to fix these issues. Sometimes, not always. So we're going to talk about the different types of mutations that can occur. We're going to focus on mutations in protein coding genes, and then we'll talk about some of the repair mechanisms and why they're so important. This will not be exhaustive. I won't even really talk about plant repair mechanisms, but they do have some different ones than we do. So let's go back to basics, right? A mutation is a change in the DNA sequence. Sometimes there are ways to repair these, luckily, but not always. The effect of a mutation is not always something that we can observe. So we need to talk about types of mutations and how they can impact things. So we think of mutation colloquially when we speak of it as, I'm a mutant now, I grow 3 arms, or I become rat boy or something like that because I was exposed to nuclear radiation, right? It's not really the case. I'm sorry to burst your bubble, but most mutations, particularly when you're exposed to a high energy radiation, lead to cancer. You don't become rat boy. So don't try it is my advice. But in many cases, mutations, the impact of them is kind of determined by where they're happening. So let's talk about the 2 terms that we've talked about previously with Mendel, but this time we're going to add in our molecular knowledge. So our genotype is the actual sequence of the DNA in the genome. A mutation is always going to change the genotype. Now, do not fall in the trap of assuming that mutations are always bad. We don't know. I will tell you how to assess the impact of a mutation in a moment. But here we can see this genotype, which was TTT, changed because of a mutation to TTA. Okay. Every time you have a mutation, a change in the DNA sequence, the genotype is going to change. Phenotype, which we know is partly controlled by genotype, phenotype does not always change when there's a mutation. This is kind of we don't know until we see the organism, right? So growing the 3rd arm, we don't know that that's going to happen. And not all mutations lead to a change in phenotype. I'll show you a classic example here in a second, but let's talk about the different types of mutations in the context of protein coding genes. Okay, we're going to talk about first what we call point mutations. Point mutations are changes at a single nucleotide, a single base. We also call these single nucleotide polymorphisms, SNPs. Now, there's different things that can happen here. Okay, you could have a SNP, a change, like here, TTT to TTA. That's going to change the amino acid in this case. We would call that a missense mutation because it changes the amino acid. You could also have a change like here we have TCA, which gets changed to TGA. That changes from an amino acid to a stop codon. That's called a nonsense mutation. I would bet that this protein is no longer going to function properly because it went from being probably 1, 2, 3, 4, 5, 6, 7, 8 plus amino acids here to now it's only 2 amino acids long. But we don't actually know. So these mutations may affect phenotype or they might not. It could be positive. The effect on phenotype could be positive. It could be neutral. Or it could be negative. We don't know. I'm going to show you how to assess that in a moment, but let's look at a classic example of where it does affect phenotype. The classic example, and we saw this in Mendel's peas as well, is albinism, where you have some sort of pigmentation and a mutation leads to a lack of pigmentation because there's a mutation in a pigment-producing enzyme that makes it non-functional. A brown-furred individual, and here is an albino individual with no pigment in there. This is a mutation that has affected the phenotype. Now, is that going to be positive, neutral, negative? Well, that's where it gets complicated because we have to view this in context, okay? We have to understand where this is occurring, and we have to assess its impact on reproductive fitness. So let's talk natural selection. This is my one chance to talk about this. If you take BIO202, They talk a ton about evolutionary fitness and things like this. This is my chance, so I'm going to have fun with it, right? So, natural selection. You've probably heard that term. Darwin presented evolution by natural selection, right? Okay, how do we assess this? Well, mutations are going to occur. We need to assess whether they affect reproductive success. In evolutionary biology, we call reproductive success, we call that fitness, okay? So you may have heard of survival of the fittest. That does not mean survival of the person that can lift the most or can run the farthest. What it means is the individuals that have the most offspring, that are the best suited to their environment, those are the fittest individuals. It's reproductive success. So in any population, there's going to be variants or strains of the organism that survive and reproduce better. We call these organisms having been selected for. That's the selection part. Things that survive and reproduce better are selected for. Things that don't survive and don't reproduce well, they're selected against. There are, of course, going to be traits that allow them to do this, and those traits will then become more common in the future. So certain phenotypes will become more common because they are selected for. This is the basis of natural selection. Over many generations, evolution will lead to descendants, offspring, that have different traits that are best suited for their environment, or they will go extinct, okay? We've seen that. We've seen that happen many times on Earth, right? The dinosaurs, they went extinct because they could not adapt to the changing Earth after a giant asteroid hit it, right? And those scrawny little mammals became the dominant animal species on land. So, natural selection. Natural selection, we can sum it up here. We have a population of mice, and there's a mutation that causes some of the mice, instead of making brown fur, to make black fur. Now, the black fur blends in very well with the environment. Let's say they live in craters in the moon, right, where it's all dark lava. So the population has two different versions of the trait. We have the light fur and the dark fur. The dark fur blends in well. The hawk is going to be able to spot the mice much easier, and it will probably eat and kill the light-colored ones. If it does that before they reproduce, the majority of the offspring that are produced by the fitter individuals will be dark-colored pigmentation, and over time we are observing evolution here. All right, this process here, we have to take it into context. What if suddenly, I don't know, there was a volcanic eruption and all of the ground was covered with light-colored ash? Well, then these individuals might be fitter in that context, and the population would shift back to light-colored fur. We have actually observed this occur through evolutionary time with moths during the Industrial Revolution. The dark-colored moths sat well and were hidden on soot-covered trees when we were burning lots of coal. And as we stopped burning coal, the light-colored moths were selected for because of this. So evolution is contextual. It's always happening, but it doesn't go towards a certain direction. It's just about which organisms survive and reproduce, particularly reproduce the best. All right, so that's my short little speech on how all of this molecular stuff fits into the larger context of biology. There's another class of mutation called a silent mutation, where you change the codon, GUU, to GUG, but it doesn't actually change the amino acid. That's why it's called silent. This should not affect the protein in any way, and silent mutations are generally not in any way bad. Okay, so those are the SNP types, the single nucleotide polymorphisms. We can also have cases where we add or lose bases. And if this does not happen in a multiple of 3, this is going to cause what we call a frame shift. So here we have a sequence where 2 bases get added. So it was AAG, TCC. 2 bases, these AT got added. So now it's AAG, TAT, CCC. You can see that this has shifted the whole thing, and the reading frame for translation has shifted Frameshift mutations often change a large number of amino acids. Again, though, is that good? Is that bad? We don't know until we assess it in its context. The same thing can happen with deletions. You delete 2 bases here, and we have a frameshift here. This one's very detrimental. It creates a premature stop codon here. All right. Whether a sequence has been added or removed, we don't actually know because we often don't, it's kind of a chicken egg, like which is the normal version, the one with the extra bit or the one missing the bit. We call these mutations InDels, insertion deletions, because we sometimes can't tell the context of, does the person missing the bit, is that the normal state, or is the person with the extra bit the normal state? So we call them InDels because of that. So if you see that term, that's what it means. All right, so here's a summary of the different types of mutations we talked about. Of course, there are more possibilities. We talked about small changes, but we looked at their ability to affect the protein coding sequence. So silent mutations have no effect on the protein sequence. Through evolutionary time, we tend to see differences between us and our most common relatives, basically, are silent mutations because they don't affect the protein, but they do change the sequence. Missense mutations, we are changing the amino acid. So here we've changed from proline to threonine. Nonsense mutations insert a premature stop codon into there. Frameshift mutations, on the other hand, are when you have insertions and deletions not in a multiple of 3. That will shift the reading frame over. Okay, so now we understand mutations. What causes mutations, right? This is something that, of course, we see in popular culture all the time. And I'm sorry, but I'm here to tell you that one of the most common causes of mutations is not ever talked about in comic books, okay? It is not being bit by a radioactive spider. It is, in fact, replicating your DNA. Errors in DNA replication are a large cause of mutations. DNA polymerase makes mistakes. And if those mistakes don't get corrected, there is a mutation that occurs. So that'll actually be an important point here in a sec. I will come back to these errors in DNA replication. We have physical agents out there that can cause mutations. Particularly, this means for us, high energy radiation. So UV, X-ray, gamma rays, okay? Gamma rays don't generally turn you into the Incredible Hulk. They tend to lead to mutations that break genes, and then that can lead to cancer. So I'm sorry, comic book fans. I'll show you how UV affects DNA in just a sec. We also have chemical agents that can cause mutations. Things like DNA binding or damaging chemicals that get in there and break the DNA in some way, or these things called reactive oxygen species like hydrogen peroxide, that technically is a reactive oxygen species that can break our DNA in ways. So these chemicals can cause DNA damage as well. There are a large number of things that can cause DNA damage or increase the chance of DNA damage occurring. Now, let's look at one specific one, UV. So you're probably aware that the more UV exposure you get, the higher your chance of getting skin cancers, right? So that's why we encourage people to wear sunscreen to cut down on the amount of UV that their cells receive. So what UV radiation does to your DNA is anywhere there are 2 T's together, UV radiation can come in and the high energy photon comes in and actually breaks the hydrogen bonds here and causes like 2 T's here to bond together. Okay, that is kind of funky because what happens is the DNA double helix creates this like bulge in there. And now this is not problematic in and of itself, but what happens when your DNA polymerase comes along? When DNA polymerase comes along to replicate this DNA, it's like, dude, what the heck is this? I don't know how to deal with this. There's 2 T's bonded together. So it kind of guesses, and it makes a guess, and it adds a random base. Over time, those mutations, those changes in the DNA sequence, can accumulate and could potentially be in cell cycle genes, which could lead to cancer. All right, so that's one of the ways that mutations can accumulate in your genome. Now, we've talked about cancer previously in terms of cell division and breaking the regulation of the cell cycle. as we've said, there isn't just one mutation or event that causes cancer in most cases. If you are the guys at Chernobyl that were going in and pouring concrete into the reactor, that event being exposed to so much high energy radiation, that of course did lead to cancer for many of them. But for most people, cancer is the result of mutations over a long period of time that get accumulated, and there is an element of random chance to this as well. The accumulation of mutations builds up basically throughout your life, and that is why we see cancer risk going up the longer you live. So unfortunately, I'm here to tell you the bad news. If you live long enough, you as a human being will probably develop some form of cancer, just because of the accumulation of mutations. Now, what causes that? Well, we do have some things that increase your risk. They increase your risk of accumulating these mutations, but they don't necessarily instantly give you cancer, right? So I know it's a common term like, oh, that's going to give you cancer. That's not accurate, right? That's going to increase your risk of cancer would be a better way of saying it. Although that's not as cool when you're like making fun of your friend who's doing something dumb. So let's look at some of the risk factors here. Smoking anything, right? Inhaling burning stuff is not good for you. Chemicals that are formed when things burn often are what we call carcinogenic. They can lead to DNA damage. We do see that excessive alcohol consumption can lead to increased risk of cancer. Alcohol is a toxic chemical. High or constant UV exposure. This is particularly important when we think about how much we're in the sun, right? And your body responds to high UV exposure. If you've ever gotten a sunburn, that's your body saying, hey, you just got a huge dose of UV. All of those cells are screwed up. We're going to just destroy all of those cells and try again, right? Because they're probably screwed up now. But the biggest factor is age. And that's because the more times your cells divide and replicate, the longer they've had to, number one, be exposed to things and accumulate mutations. But every time they replicate, there's a chance DNA polymerase makes a mistake. And if that mistake doesn't get corrected, you're accumulating mutations in those cells. So your own DNA polymerase just living and existing is a risk factor for cancer because it makes mistakes. I know that's a little bit depressing, but don't worry. You do have some repair mechanisms in there. I'm going to talk about two repair mechanisms and like the nuclear option here. So I guess let's start with the nuclear option. Programmed cell death. We've talked about this previously, apoptosis. That's like when you get the sunburn, right? Your body decides that, hey, those cells are way too damaged. We're just going to destroy those cells to prevent uncontrolled cell growth there. Okay, that's there's no going back here. There are some other repair mechanisms like base excision repair, where we remove damaged DNA bases and fix the issue. And then there's one called this methyl mismatch repair. This is really interesting. We're still learning about this, but old DNA strands get methylated and we can mark it and know that that's the old strand and use it to fix a new strand of DNA that's being copied when errors happen in there. So we like, hey, that's the master copy. We're going to fix the new one here. All right, I'm going to show you an image that shows you just how much DNA damage your body actually repairs. So I'm going to warn you, this is slightly disturbing. This is a child with xeroderma pigmentosum. Here, this child, who is probably, I don't know, we're talking 4 or 5 years old, they lack DNA repair genes. They're lacking the normal DNA repair genes. And you can see that even through minor exposure to UV, they are developing skin cancers and growths there. Now, this individual, they know they have this, right? They avoid the sun as much as possible, but they are still getting this. So what I want you to take from this is that your body repairs a ton of DNA damage. And if you don't have those repair mechanisms, you would develop cancer very, very early in life. All right, so let's look at 2 ways of repairing things. Base excision repair is when we realize that there's been an error. Okay, so here's a damaged DNA base. Well, here, the first thing that happens is an enzyme comes in and actually cuts out the damaged base. And then it starts chopping back a little bit extra. DNA polymerase 1, which we've talked about previously, which does some proofreading here, comes in and fills that back in and seals up the gap with DNA ligase, and we've fixed the error that is in there. Great. That happens all the time. This base excision repair is important in fixing damaged bases. This is one of several ways that DNA can be repaired. Plants, if you go on to take upper level genetics classes, they talk about photoreactivation repair and things like that. Plants kind of have it bad. They have to sit in the sun, so they're constantly exposed to that high energy UV radiation. So they actually have mechanisms of repairing their DNA that we don't have because they have evolved a different lifestyle than us. Different type of repair is called methyl mismatch repair. You can see here that this DNA is getting replicated. So the blue strands are the old parental strands, and they actually have special marks on them called methyls. These methyls basically signify that these are the old strands. They're the master copy, right? And you can see here that the new strand that was being made, there's been a mutation here. We had a G there, but a T accidentally got added, and it's not fitting quite right. So how does the machinery know that this green strand is the new one and the blue one is the old one? Well, it looks for these methyl marks. This new strand doesn't have the methyls on it. So it goes, all right, I'm going to refer to the old one and use that to fix this. And now we have a GC here instead, which is fixed. So that is a clever mechanism cells can use to understand which template to use to fix the DNA. All right, so to finish up, a mutation will always affect the blank of an organism. Go ahead and pause the video. Think about this one. Okay. Hopefully, you remember that it will always affect the genotype because a mutation is literally a change in the DNA sequence, and the genotype is the DNA sequence, right? Whether that affects the fitness, how well they reproduce, whether it affects the phenotype, that remains to be seen. So we can't tell just by looking at the mutation what is going to happen in terms of phenotype or fitness. All right, so hopefully understanding the genetic code puts this all into better context, although I know it's not cut and dried, right? Mutations are not always bad. That's strange to think about because of how we talk about mutations normally. So a mutation is a change in the DNA sequence. That's all it is. This may or may not affect the phenotype. If it affects an organism, it could be good, right? It could be a good phenotype. It could be bad. It could be neutral. We don't know until we examine it in context. So mutations may affect fitness. Fitness is reproductive success, right? This whole thing is the basis for natural selection. Our types of mutations that we talked about SNPs, single nucleotide polymorphisms. We had missense mutations. Those change the amino acid sequence. Nonsense mutations introduce a stop codon. Silent mutations change the sequence, but the amino acid is the same. It still codes for the same one. Insertions and deletions add and remove bases. If it's not in a multiple of 3, that results in a frame shift there. Errors in DNA replication made by DNA polymerase, they can cause mutations, and that's one of the key places that they happen for us. We also have chemical and physical agents that lead to mutations. How many mutations happen because of this? Well, when we look at parents and offspring, we see in a human genome like somewhere around 300 mutations. So not an unheard of rate, but that's 300 out of 3 billion. That's not a high rate, right? So DNA replication, pretty good, pretty safe, but it does make mistakes. Cancer is caused by the accumulation of many mutations in cell cycle genes. So over time, your chance of cancer, your risk of cancer goes up because of the accumulation of mutations. We did see that there are ways to repair mutations. We have DNA repair mechanisms like base excision repair, cut out the damaged bases and refill it in, or that methyl mismatch repair where it uses the old strand to fix a new strand. Then we have the nuclear option, right? Apoptosis, programmed cell death if the cells are too far gone. All right, that's it for our understanding of the genetic code.